All right, welcome to this explainer. Today, we're doing something a little bit different, and frankly, it's going to completely shift how you tackle your Cambridge AS and A-level biology exams. We aren't just doing a standard biology review today. No, we are literally going to learn how to think exactly like an examiner. By breaking down these visual blueprints, we're going to transform your whole approach from just, simple memorization to absolute strategic mastery.
So let's kick things off with a pretty painful realization. It is honestly heartbreaking to see students fail, not because they don't know the science, but because they walk right into the examiner's traps. I mean, think about it. Why do 9 out of 10 students lose vital marks on simple calculation questions, even when they understand the biology perfectly? Well, let's find out.
Okay, imagine you're doing an ATP respiration calculation. You calculate the respiration rate perfectly. You get 4.5, and you move on. But wait, you completely forgot to write down the unit. That one simple omission instantly destroys your score for that question. It's what we call the unit trap. But there is a massive safety net here. If you actually showed you're working, the method mark lifeline steps in to save your grade. Seriously, even if your final answer is totally wrong, just showing your exact procedural steps secures those process marks. It is an absolute game changer.
Okay, moving into Section 1, Core Biology Concepts and Mastering the Fundamentals.
To completely dodge those fatal traps we just talked about, principal examiners say you need a bulletproof systematic framework for every single question you answer. Here's your 4-phase framework for high-mark answers. Step 1. Identify the core biological concept. Step 2. Extract specific evidence and data from the provided stimulus. Step 3. Apply scientific reasoning using logical, causal statements. And finally, Step 4. Finalize your answer using exact syllabus terminology. Let's actually see this in action.
When you apply that 4-step sequence to a triglycerides question, you map the molecular structure breakdown directly to those steps. But you've got to watch out for the terminology trap. This is where students lose points by using generic, near-miss vocabulary instead of precise biological terms. Vaguely mentioning bonds just isn't going to cut it. You have to specifically identify ester bonds when you're talking about lipid components. In these exams, specificity is literally everything.
Next up, Section 2, the Technical Methodology Toolkit for Success.
Now, let's talk about processing commands and analyzing data under extreme exam pressure. The crucial difference here is between a flawed generic description and a top-band evaluative answer. A flawed answer usually just recites textbook theory without ever actually looking at the data provided in the question. A top-band answer, on the other hand, actively pulls specific data points from the stimulus and maps them directly to biological principles using precise command words.
For example, when looking at transport in plants, examiners explicitly warn us about the superficial breath trap. Top marks require analytical depth. You absolutely must distinguish between the command word describe, which is simply stating what is happening using specific values, and explain, which demands that you detail exactly why it is happening based on the underlying theory. Mixing those two up is a major mark killer. And that brings us to a huge examiner red flag: vague contextualization. Examiners constantly report that student answers are just way too generic. If you want to hit those highest grade boundaries, your responses must be tailored specifically to the exact context, the specific data, or the exact species provided in the exam paper. No exceptions.
Let's look at a heart wall thickness case study. If a student just slaps down a simple ratio of 2.5 but provides absolutely no units and shows 0 methodology, they get 0 method marks. It's brutal, but true. Top-band marks require targeted, logical reasoning applied specifically to the context. You need to be comparing atrial versus ventricular walls with exact evidence-based reasoning.
All right, let's make this interactive. I'm going to give you a 10-second challenge. I want you to think like an examiner for a second and see if you can spot the single fatal error in a student's calculation before I reveal it. Get ready.
Did you catch it? The student calculated the mean increase perfectly as 15.5. But they completely forgot the millimeters unit. It is a classic, agonizing error. And right alongside that is another massive pitfall: confusing antibodies with antigens. Remember, an antigen stimulates the primary immune response. An antibody is produced in response to it. Examiners see this confusion constantly, so always define your terms clearly to show absolute mastery. Moving right along to Section 3, Critical Analysis and Evaluation. As we push into higher-order assessment objectives, those critical A-star differentiators, elite exam strategy requires rigorous statistical precision. You have to know exactly which statistical test in your toolkit to deploy. Are you comparing means between two sets of data? You need a student's t-test. Analyzing categorical frequencies, that's chi-squared. Identifying relationships between variables, you'll use a correlation test. Nailing this is a huge factor in complex topics like biodiversity and ecosystems.
Taking a deep dive into the link reaction perfectly exposes how this critical analysis is actually tested. You cannot just write a messy, vague statement like pyruvate breaks down. No way. You must execute clear notation. You have to explicitly state that pyruvate is decarboxylated and dehydrogenated, fully justifying your conclusions. It's all about linking cause and effect without stumbling over terminology.
All right, Section 4, evading the terminology trap and keeping specific context. Let's tackle one last major hurdle before our recap. This is truly where the highest achieving students separate themselves from the pack.
A really common, fatal error in data